So, hi everybody. At first, I'm very glad to have so many participants to this third event. I hope we will be able to have good deals together. And so I will try to explain you what will be the following talks. We'll have a nice talk from Laurent Philippot, which is a plenary talk. This talk will introduce all the session. And then after, I will give you a tour of the 4PMI platform, which is a platform devoted to plant and microorganism interactions. I'm sorry for those that were already present in the previous event, so there might be some redundancy. And after that, we'll have three talks to illustrate some results issued from the SolACE project. So there will be a first talk from Stéphane Declercq, which is called the infinitively small at the service of humanity. And we need right now that we need many services for humanity. The second talk will be from Sarah Shivanshik, and the title is Impact of Farming Practices Shaping Microbial Communities. It's also a talk which is issued from results of the SolACE project. And the last talk will be from Germain Montazeo. It's more a plant-to-plant interaction focused talk. But I'm pretty sure you will like it. And the title is Root Trait Diversity and Early Growth in Wheat Varietal Mixture under Contrasting Nitrogen and Water Conditions. And then after, we should have a further question and answer discussion of roughly 45 minutes. So before leaving the floor to Laurent, I would like to again remind the speakers to be tight on the length of their talk. Okay. So Laurent.
Thank you, Christophe, and thanks you for giving me the opportunity to talk about the importance of microbes for plants. I will share my screen now. Here it is. Is it okay? Is it in presentation mode?
Yeah, perfect.
Perfect. So the title of my talk is Going Back to the Roots, the Plant Microbiome. As you know, plants are hosting diverse and complex microbial communities, and these communities are different between the endosphere, the phyllosphere, or the rhizosphere. These microbes are not just there. They can help plants in many ways. They can help plants to cope with various stresses, such as cold stresses, drought, flooding, osmotic stresses. They can help plants to fight pathogens. And they can also help plants to cope with nutrient limitation. And this is a famous example of symbiotic nutrient fixation, in which Rhizobia, a bacteria, is living inside the roots of legumes and is fixing atmospheric nitrogen and giving it to the plant for its nutrition. And this is a figure from a paper published in Science some years ago. So this is a figure, a very simplified nutrient cycle. And in green, you can recognize the green arrow here. Can you see the mouse? Perfect. The green arrow is nutrient fixation. I just talked about it. And this is how the nutrient cyclings look like before. This is how it looks like now with the Haber-Bosch process, which is the industrial fixation of atmospheric nitrogen to make fertilizer. And this process is not so important, but it's in the same range as the biological nutrient fixation in atmospheric system, in terrestrial system. And it is estimated that about 1/3 to half of the nutrient in our bodies come from this process, come from the Haber-Bosch process. So the problem is that we are using too much nutrient fertilizer, and the cost of nutrient fertilization in Europe only is estimated between 70 and 320 billion euros per year. So in this context, this process, symbiotic biological nitrogen fixation, is very interesting because, again, the bacteria here is fixing nitrogen, atmospheric nitrogen, and for this purpose is using the carbon provided by the plant because this process is very costly. It requires a lot of energy. This fixation is done by the enzyme called the nitrogenase, and then the bacteria give a fixed nitrogen to the plant for its nutrition. It looks very simple on this graph, or rather simple on this graph, I would say. And it's actually a very complex process, which involves like a communication between the plant and the microbes, and also the formation of a new plant organ, the nodules on the roots of the legumes, and the bacteria is fixing nitrogen within this new organ, the nodules, and you can see example of nodules on these figures on the right side on the roots. So it's very important because, as they say in this paper in Science, it's the route to self-fertilizing plants. Plants, they don't need fertilizer. And as I explained, fertilizer is not so good when it comes to the environment. There have been two main lines of research during the last decades, so that plants, crops, or cereals other than legumes could benefit from the biological nitrogen fixation. One way was to create new partnerships so that other bacteria and rhizobia could fix nitrogen and supply this nitrogen to maize or wheat, for example. Or another way was to introduce this bacterial enzyme, the nitrogenase, into plants. This is one recent example of this line of research, the first one. But in this case, they transfer the nitrogen fixation genes, the entire cluster, from Klebsiella into endophyte living in wheat or in corn, such as Pseudomonas. And you can see in this figure here, for example, that the level of transcription in this new host is the same as the one in Klebsiella. They successfully managed to transform this capacity into endophyte of crops, cereals. And this is a line of research going on for decades. So let me now introduce you to this giant landrace of maize growing in the Sierra Nevada in Mexico. So it's a very tall maize. I think it's up to 5 meters high. And it has these aerial roots producing a lot of mucilage. And it's growing on a very nitrogen poor soil. And what's interesting is that this mucilage is full of carbohydrates. It contains a lot of carbohydrates. And high nitrogenase activity has been observed in this mucilage. As you can see, the level of nitrogen fixation in the leaf, the stem, the roots. The roots without the mucilage are very low, but there's very high nitrogen fixation activity in this mucilage. And this is possible because this mucilage is protecting bacteria from the oxygen, because nitrogen fixation is very sensitive to oxygen. And the bacteria are also using this carbon from the mucilage as energy source for nitrogen fixation. And what's even more interesting is this: when you look at the nitrogen in the plant coming from the atmosphere, you can see that it was a field experiment. During these 3 years, between 40 to 80% of the nitrogen in the plant comes from this nitrogen fixation by bacteria living in this mucilage. So it means that this maize living on nitrogen poor soil managed to do through evolution alone what we are trying to do as researchers for decades, to develop a symbiosis with nitrogen fixers. This is in orange nitrification. This is the oxidation of ammonium into nitrate. And this is also an important process when it comes to nitrogen availability, because nitrate is less stable in soil than ammonium. So it can lead to leaching, and then leaching can lead to eutrophication, as you can see on this picture. And the produced nitrate can also be lost from the biosphere by denitrification. It's transformed back into gas. So nitrification is leading to a loss of nitrogen for the plants. That's why we are now using nitrification inhibitors in agriculture. And you can see on this table that the total tonnage bound here in this last corner, that this is the amount of nitrification inhibitors that can be purchased without notification to the authorities in Europe. And this is ton per annum, and this is a growing market, as you can see. So we are trying to limit this process in agriculture. So now let's go to the tropical grass savanna. This is Hyperrhenia savanna, and in this study, for example, they observed that nitrification, this process, was lower next to the root of this grass, Hyperrhenia, compared to the surrounding soil. And this is also what you can see on this slide, where you have a very nice negative correlation. When there's a higher root dry biomass, you have lower nitrification rates. And I mean, this is a line of research that was conducted for decades. And in 2009, they finally found the mechanisms is that the roots of these grasses are producing compounds, secondary compounds, which are specifically inhibiting the oxidation of ammonium into nitrate. So it's blocking nitrification. And this is an example of compound, which, in this case, from this grass, which is called the Brachylacta. So this is very good, because when the nitrogen is in the form of nitrate, and it can decrease the N2 emission. And this is what you can see here. This is a field experiment, which was conducted for 3 years. So they use different types of these grasses with increasing amount of biological nitrification inhibition, so BNI. So increasing from 3, this is arbitrary unit, from 3 to 53, more or less, and with a control and the soybean as control soil, plant free, and soybean as control soil. So what you can see here, that the ammonia oxidation, the first step of nitrification, is decreased with increasing biological nitrification inhibition. And what was also interesting is that the N2 emission, and N2, if you don't know, it's an important proton greenhouse gas, the N2 emission are also decreasing with this increasing BNI activity. So we have been looking at widespread this process, this plant trait was, and it's not so common in crops in cereals, and this it's not in wheat. There's a paper published last year. They found something in maize, but it's to be confirmed. So this is a paper which was published last year in PNAS. So what they did, they transfer the chromosome region that control for this biological nitrification inhibition in a wild grass, Lemus racemus, which is a wild relative of wheat. They transfer it to elite variety of wheat, the Muniol varieties. And what you can see here is that, so this is, whoops, sorry, a bit too fast. This is the biological nitrification inhibition Muniol varieties resulting from the transformation. You can see that there's more ammonium in the soil compared to the control wild type. There's less nitrate, and there's a decrease of nitrification. So this transformation works well. And what's even better is this one, is you look at the grain yield in ton per hectare. You have an increase of the grain yield in this new cultivar, this modified cultivar, And this increased yield is even stronger when there's no fertilization here. So this is the one with the control, and this is the one with this new plant trait. So what I'm trying to highlight here, it's this natural ecosystem. Biotic interaction are very important for plants. And this is not as strong in agricultural system. And one of the reasons is that domestication was detrimental for plant microbes interaction, because simply microbes were not taken into account during the breeding process, and plants were grown in systems with high nutrient and pesticide input. So there's a new kind of paradigm shift last year, like moving from plant breeding to the holopiont breeding, so like breeding the plant and the associated microbes, and even not to breed only the associated microbiome. And I will explain you this with this slide. So you grow your plant with the microbes, and then as a classical breeding process, you select the plant according to phenotype trait that you are interested in. In this example, it's the number of flowers. So you select the microbiome from this plant with, in this case, with more flowers. You are planting a new generation of seeds into the sterile soil, and then you inoculate the very same plant then here with this microbiome coming from this plant that you selected. And then you repeat this. So you're selecting only the microbiome, you're not changing the genetic pool of the plant. And this is an example of this kind of approach, which was conducted in Dijon in the high throughput phenotyping facilities that Christophe will talk about. So in this case, we use Brachypodium distachyon as a model plant, and the leaf gridness as a proxy for plant performance. So it's like high or low leaf gridness. And the experimental design, it was a 10 generation experiment and involving about 8,800 plants at the end. So these are ordination showing how similar or different microbial communities were during generation 1 to 5 and 6 to 10. So the color code is like the blue dots is the control, microbial community coming from the control plant, so random selection. Yellow orange, that's microbial community selected from the plant with a low leaf greenness. And greenish color, that's the microbial communities coming from the plant, selected from the plant with a high leaf greenness. And you can see that during the 5 first generation, the green, the yellow, the bluish dots are together. There's no selection of the microbial community. But during generation 6 to 10, you can see a nice separation. You can see that the communities from the control plants in blue are different from the communities from the low leaf greenness plant and from the plants with a high leaf greenness. Okay, so we did actually manage to select the microbial community, and we did observe also, and it's the most important, an effect on the plant phenotype. So this is the leaf greenness ratio compared to the control along the 10 generation. And you can see that it's more clear after generation 5, so it fits with the microbes, but you have a significant increase in the leaf greenness for the plants. And this is the same results according across generation with a high leaf greenness and a low greenness plant. So it was the same seed all over experiment, we just selected microbial community. And I think I was too fast actually. You put pressure on me, Christophe, because now this is my last slide. And this is just a slide to show you that harnessing plant microbes interaction is a very hot topic for the last years, and this is not about to stop. Thank you for your attention. I hope I convinced that of the importance of microbes, in case you are not already.
Well, thanks a lot, Laurent, and no problems to go faster than planned, because we'll have room for more questions later on. Well, there is not so much question on the chat, so but maybe we could admit people asking question already by turning on the microphone if they ask for. Myself, I have a first question, Laurent. When you see your two papers, the one where they engineered the bacteria to help them to do symbiotic nitrogen fixation with cereals, and the second one, yeah, the previous one, this one, and the next one, I was asking myself, well, as you quoted, it's since a long time we are working, many people are working on association between cereals and bacteria. So first, stupid question, why did we lose that? And second question, how we could try to go faster now to engineer them either from the side of the bacteria or from the side of the plant to get such a partnership between plants and microbiome, to have a good deal?
Oh, tough questions. Well, I think at first we didn't look at the right place. It's like this example that I showed, the two other example. It's like it was plant grown in very naturally poor environment, and we are more focused on the elite cultivars or varieties and one performing well, and we are not interested into those. So that's, and it's actually this plant growing on very poor natural soil. It's where this kind of interaction is more likely to happen. So I think that during many years, we are not looking at the right place, if I may say. And what was the part of the question?
What could be the perspective of engineering plants, elite cultivars, either from the side of the plant or from the side of the bacteria, and trying to modify the bacteria, as in the papers that you are just showing, trying to get rid of the inhibition by nitrate and so on, and trying to have the plant delivering nice exudate to the selected to the engineered microbia. Is it promising or not? Is it feasible at a large scale or not?
Well, it is for the nitrification inhibition. It will be easier. The problem with what the example I show you is that we are talking about GMOs, and this depends if you are in Europe or on the other side, if you can use that, because the plants, and this is kind of, it's a elite cultivar of wheat, so this one is already could be used tomorrow basically, and the results are there. But then it's more like a regulation problem. For the nitrogen fixation, then there's another dimension also about regulation, is you need to inoculate microbes. In this case, you will need to inoculate this modified endophyte. And again, it's these microbes are GMOs. So in both cases, you have to deal with GMOs. There's also a line of research, and they are trying to breed this, when it comes to this nitrification inhibition, they are to breed this wide relatives of wheat having this capacity with this elite cultivars, but it takes more time, as you understand. But in this case, you will not have this problem with regulation and the use of genetically modified organisms.
Okay, thank you, Laurent. We have some questions on the chat. The first one comes from Sarah. She's asking if you could explain a little bit more how you select the microbial community and how you transfer them in practice.
Okay. Go to first. How you select them? I mean, you don't select the microbial community per se in this case. I mean, you select based on the plant traits. So you're choosing the plant, in this case, having the biggest flower and the more flowers. And then we are collecting the microbes from the root system. So we are collecting the rhizosphere and we are washing the rhizosphere to collect the microbes. And this is kind of this soil suspension coming from the rhizosphere, which is used for the next generation. So we're not isolating the microbe, it's more like a soil suspension, a rhizosphere soil suspension, which is used to inoculate the sterile soil with the seed.
Thank you, Laurent. Two more questions. The first one, I think I could answer too, but a small question from Prakash Gimir. I hope I spelled well your name. Is it possible to classify the root microbiome interaction phenotypes based on the root structure and typology and topology? I would say no.
I think it will not be enough because roots are producing a lot of exudate, and these exudates are very important also when it comes to plant microbes interaction. So it's not only the morphology of the roots. This will, I don't think that this would be enough. Morphology
of the roots is one key point because it could trigger some specific colonization of the soil, but the key is the exudates. You're right. And the last question, because I think after we'll have to keep on time. There is another question from Prakash. From Borbola Biro, is microbial denitrification inhibitors selecting the microbial community? Is it really acceptable? Do those compounds affecting on the biological nitrogen fixation organism?
It's a very good question, actually. It's not because they are produced by the plant, but it's because, I will rephrase it. It's not because the compound is natural, but it's good. So in this case, actually, I'm writing another paper on that. It's like not enough studies has been done, in my opinion, to test the effect of this nitrification inhibitors produced by plant on non-target microorganisms. And even for nitrifiers, they are focusing on, even for nitrifiers, they are focusing on bacteria, and there are other types of nitrifiers from Archaea or Nocomamox. It's a new group of nitrifiers which was capable to do both steps, and it was published in Nature 2 years ago. So we don't know, and I think we need more kind of ecological studies assessing the effect of these nitrification inhibitors on nitrifiers themselves, but also on non-target soil microorganisms.
Okay, thanks again, Laurent, and thanks for the audience. Now I will show you some the facility that Laurent was talking about, which is called 4PMI, which is in Dijon. I will share my screen. I'm really sorry for the people which are very studious and which were already present in the last event, and I'm grateful to them, in fact. I hope you see. Can you see my screen? Can you see my screen? Can you see the screen?
Yep, you're all set.
Okay. Well, I will make a little tour of the platform we have in Dijon. So it's called 4PMI. I hope you see the upper of the screen. 4PMI stands for Platform for Plant and Plant Microorganism Interactions. I cannot. Okay. So the first general slide. What are the expectations from high throughput phenotyping? The first one is to help to characterize numbers of genotypes, mutants, to discriminate them for a range of species. I put some example at high resolution, dynamically and non-descriptively, because this is what we are working with when we are working with large numbers of genotypes or ecotypes in quantitative genetics or in genome-wide association studies. And to do this in a variety of environmental scenarios, for example, those of climate change or others, and in a variety of interactions with plant-plant interaction or plant microorganism interactions. Second objective is to measure agronomic traits issued from the image, and by agronomic traits, I mean agronomic relevant traits, because it can be either morphometric traits, for example, the depth of a root or the area of a leaf, but also the function which is realized by these structures. And lastly, we use most of us use this data to feed models, various models, for understanding and simulating the plant functioning according to climate condition. In fact, we are able, using the combination of modelization, phenotyping, and genetic resources, to gain a characterization of the mechanism, the relation with the traits, and the relation of the traits with the performance of the plant. So I'll make a tour of the platform. So in Dijon, we have a platform which has 6 environmental conditions, and plants are automatically conveyed. They are grown in pots or rhizotubes, and they are conveyed from the various greenhouses. As I said, we have 6 greenhouses to 5 phenotyping cabins. You see the plant entering the cabin. In the cabin, you will have various cameras phenotyping the plants in various wavelengths. So we have a capacity of 1,800 pots and 1,200 rhizotubes. And we can phenotype the plant in near infrared, in visible, and in visible light, in near infrared light. And we make the plants turn in the different cabin using robots, and this dynamically, to extract the forms from the shoots. This is what you can see right now. So the age, the diameter of the stems, the number of leaves, the area. So to do form extraction. But we can also use our 1,200 rhizotubes, which are automatically irrigated with various nutritive solutions, to phenotype the root system. At the beginning of the experiment, we are often using germination chamber to speed up the germinating process. And then after, we phenotype the plants in two kinds of cabins. The first one is a high resolution, low, medium throughput cabin. This one, 7 micron resolution, which is very relevant for looking at fine roots or even root hairs. And we can gain images such here, where you can see a pea growing with or without soil mineral nitrogen. That is to say, without or with symbiosis with bacteria. And here you have another example of what we get with a high resolution cabin. So you have progressive focus on the root system, and you can see the level of precision we have for looking at nodules. You have the same for an alfalfa plant, and you see progressively the dynamics of root prospection. And again, for a pea, you can see the progression, the growth of the roots and the nodules. In the last slide, it was pretty fast, but you could see, you could have seen also that we can even see IFA from mycorrhiza. The second cabin is a high throughput phenotyping cabin, which allows us to phenotype 1,200 rhizotubes per day in less than 5 seconds per wavelength at a quite good resolution, because it's 42 microns, which is not something you could get with X-ray or MRI. And with this, we worked on many species to work on the inter and intra diversity of the root system of the phenotype. And we use this fancy structure to equipment to work on many topics that you can see here, nitrogen efficiency up to associations. So I will now show you some additional tools we have. So at the end of the experiment, we have to get back to the reality. So we have to harvest the plant. So we harvest the plant very quickly using such a tool like this, which will be automatized pretty soon. And then you can have access to various parts the root system. I did not detail the rhizotube because I guess that most of you know the rhizotube, but if you want me to go back on this, I can do it later. And then after, you can extract some parts of the roots, and you can do any omic analysis you want, okay? But on the other side, we can also do fluxomic for C13, N15 nitrogen, whatsoever gaseous or in solution to work on fluxomics. So we have both solutions. We have the function using fluxomics, and we have also the morphometry using the 4-PMI. So this allows us altogether to have a kinetic analysis of food development. Some for the image analysis, thanks to Michael Lambert, who is assisting this workshop, we go from the original image through segmentation to gain various traits. But as we have thousands of images, we add to speed up their analysis, which was done developing intelligence artificial methods such as deep learning. This is done in collaboration with private companies and private partners like as Bastien Billot from Rouvier, but also in the frame of the Phenomorphosis project, in which with David Rousseau and Michael, we are working in more depth in image analysis through deep learning. So what are we doing with all these fancy tools? You have an example first of what Corentin Mallard, which is a PhD student in the group, did. You probably know that there is a large genetic variability of food architectural traits for soybean. So what he did, he selected some soybean genotypes, and you see that indeed there is a large variability in the root architecture. And using all the tools I was just talking before, he did some analysis of the genetic variation of the root architecture trait, and also of the functionality of these traits, that is to say, the symbiotic nitrogen fixation. Okay, so what he gained is what you see there. He already saw some nice morphometric differences. As an example, here you can see how they are differentiated in the number of nodules, which is a key parameter of the efficiency of symbiotic nitrogen fixation, and on the flux of nitrogen which enters the plant. But he did also using measurement of the content of nitrogen in the plant, some functional characterization of the efficiency of the symbiotic nitrogen fixation in the plant. So altogether, you see that all these tools, again, can be used for root and nodule structural and functional traits, as I said earlier, and this can be used towards breeding programs, but also for other purposes. And as an example, in I skip the slide, in fact. Right. So I will show this time another example that was acquired in the frame of the project INRAE GRASP by Virginie Bourillon. So she used the platform to phenotype nodulated roots of a collection of 336 P genotypes crossed by 28 rhizobia. And you can see some extract of the results where you have a large variability of the root system architecture. So this shows this again. This can help to characterize the one which will be the more efficient in performance, and in fact to go towards breeding P varieties which have an improved symbiotic nitrogen fixation, but also an improved performance. The last example, because I was talking of rhizobia, but as Laurent said, there is, and as you all know, there is not only rhizobia in the life. So the last example comes from a study from Marion Prudent. So Marion did some experiment assessing the importance of the microbial diversity. As you might know, there is a consensus saying that the more diversity we have, the more performance we should have. This holds for plant species, but it can be also the case for microbiome. So what she did extraction of the microbial communities from the soil, and then she diluted this microbial community, and she re-inoculated this microbial community in an active soil, and so she get high or low microbial community. And then in parallel, she had the nice idea to use either the wild type frisson, which is a genotype which realizes mycorrhiza and nodulation, and also she used a mutant which is not doing any symbiosis. To go fast, after a first stress, she saw nothing concerning the impact of the microbial diversity on the plant resistance to drought. It means at this stage, whatever the plant establishing symbiosis or not, there was no impact. But later on, when she looked at physiological maturity, she observed that the more the microbial diversity is, the better is the resilience to the stress. And this holds for both frisson and the non-symbiosis establishing mutant P2, which means that in fact we are often focusing on rhizobia, but rhizobia are not the end of the story. Non-symbiotic communities plays a huge role. I hope I'm still on time. So to summarize, I draw a conceptual scheme where I was thinking that we could all work on special topics, for example, the transfer of nutrients, from the shoots to the roots, and looking at the variation of these flows and the signals which are involved for various species panel and various stresses. Also, the plant and microbiome modified through exudates. We are talking on that, and you will see a speech later on which has some illustration of that. So what are the plant-soil interaction modification delivered by this? What is the benefit for plant nutrition? And when you are working on plant-plant interaction, and you will have an example in the last talk also, what are also the benefits for both plants? Well, to conclude, what can we get using high throughput phenotyping for this project? This project associating high throughput phenotyping contributes to help us to get the physiological mechanisms of the desired root traits, which are important for us. And they help us to get root characteristic, to get functional and structural traits, and to exploit the genetic variation in these traits. Using high throughput phenotyping and omics, we can analyze the molecular basis of the efficiency of the traits. We can develop markers and genetic prediction tools for breeders. Microbiome structure and function and host regulatory mechanism are key for the future of agroecology and for sustainable agriculture. And another key message is that for this, high throughput phenotyping provides precise and efficient screening of large number of plants, hundreds to thousands, to help us to take into account and to benefit from positive interaction between the plant roots and the microbiome. And lastly, altogether, I would like to highlight, I'm sorry, but I'm preaching for my powers. The high importance of the root phenotyping for faster generation of variety which have an improved performance for food production. I hope I was on time. Amelia, was the case? Yeah.
Just in time, perhaps.
Okay. I'm sorry if I was long.
No, it was good.
We see later on, but if you have questions, we could gather questions in the last session after the free talks. And now I will give the floor. So I have to stop my sharing. So I will give the floor. No, I still have the floor because Stéphane Leclerc has the first talk. So Stéphane, are you ready?
I'm ready.
I have to find your talk, sorry.
Yeah.
I have many. Well, if you have questions, you can put them on on the chat. So can you see my screen? No, it's the same always. Partager l'écran. Can you see? Okay.
Yes. Okay. Thank you, Christophe, and good afternoon, everybody. So the title here of the talk is in fact related to a short film that Christophe will show you in a few minutes. So of course I will not discuss in a few minutes about the infinity small at the service of humanity. I will simply show you a number of results that we obtained in the SolACE project. Next slide, Christophe, please. Just. Yeah, so simply to show you, of course, I'm working on the microbiome, on the microorganism. So the rhizosphere is, of course, something which is extremely important. Rhizosphere, which is the zone of influence of the roots, which is more or less a few millimeters around the root system, the root hairs. It's also named the hidden half. It's this part of the roots where the plants. Where the roots interact with numerous microbes, bacteria, fungi, for instance. These microbes can be beneficial, nitrogen-fixing bacteria, bacillus, pseudomonas, for some of them, but also can be detrimental pathogens, for instance. Note also very important that this rhizosphere contains 100 to 1,000 more microbes than in the bulk soil. So it's really the place where exchanges, dialogue exists between the plant and the organisms. Another interesting data is globally the soil contains 4 to 5 10 to the third microbial cells. It's the second biggest reservoir of microbes on Earth. And so just to continue on this slide, you see the rhizosphere, which is the zone of influence of fruit. We have also what I call the mycorrhizosphere, which is in fact the zone of influence of the roots colonized by mycorrhizal. This is basically the topic of our research team. And as you can see on the slide, on the image on the left side, well, the fungus develops inside the roots and then explore the environment extending 20, 30, 40 centimeters away from the root. And this mycelium is able to take up nutrients, take up water, to transport these nutrients and water to the plant, but also to protect the plant against a number of stresses and diseases. And so in SolACE, we were interested in this mycorrhizal fungi. And we had the first big experiment, next slide, please, which deals with the assessment of AMF colonization and diversity in the durum wheat field. I would like also to acknowledge here Marcel Doubel, which is the person that did all that work, very heavy work, and thank you for that, Marcel. So this first experiment was conducted in Foggia, so in Italy, in which 215 genotypes of durum wheat were analyzed for AMF root colonization. And this is the result in this graph. And as you can see, all the wheat genotypes are colonized by mycorrhizal fungi, which is the first interesting aspect. But the second point, which is even more important, is that the colonization rates may deeply vary between the genotypes. From the left side, you have some genotypes that only contain 3% of colonization, while on the right side, it can grow up to 50% of colonization, which also show the importance of the genotypes for their colonization by mycorrhizal fungi. This can also be an interesting approach for breeding processes for genotypes which are maybe more adapted or more conducive to colonization with mycorrhizal fungi. So on this graph, you see also that a number of histograms are in yellow and green. The yellow histograms on the left and on the right are in fact tolerant or sensitive genotypes to water stress. And so we selected 15 of these genotypes. And in the second experiment on the next slide, we measured, so we selected 15 genotypes from the field just before, and we conducted a pot experiment with mycorrhizal and non-mycorrhizal plants. So these 15 genotypes were either colonized by an AM fungi or were not colonized. And then we evaluated the AMF colonization percentage, but also the relative mycorrhizal dependency. So the relative mycorrhizal dependency is a measure of the response of, next slide.
Oh, sorry.
Is a measurement of the, no, not this one, just before.
I'm trouble with the arrows in my.
Okay, this slide, yes. So it's a measurement of the benefit that mycorrhiza bring to the plant in terms of biomass increase. And as you can see here, you have huge difference between the 15 genotypes. A number of genotypes even have a negative relative mycorrhizal dependency, which means that their biomass was decreased in presence of this specific fungus, while other on the right side of this figure had an increase in biomass. So the responses are also genotype specific. And then we measured the ratio between root and shoot and tried to make a correlation with the relative mycorrhizal dependency. And we observed a significant correlation between these two parameters. So a negative growth response, negative growth RMD, can be linked to a reduction in root biomass in presence of AMF, while shoot biomass remain relatively similar. If we take the example of SDW 4030, just below on the left, well, you can see that there is a decrease, a 35% decrease in root biomass in the presence of mycorrhizal fungi compared to its non-mycorrhizal treatment. However, for this specific genotype, shoot biomass remained relatively the same under both treatments. And this means thus that AMF induces a decrease in root biomass, but also can compensate for a decrease in root biomass. And in the third experiment, next slide, please. It was the objective to see if mycorrhizal fungi could help plant to cope with drought stress. And here we selected two. This is a still ongoing experiment, so I have no results on wheat for the moment, but the concept is that we have a circulatory system in the middle of the image. So the plant above receives nutrients and water, and then we measure, we follow three different water regimes: a water regime in the field capacity, half of the field capacity, and a quarter of field capacity. And we measure in mycorrhizal plants and in non-mycorrhizal plants how the plant responds to these three water regimes. So this is still ongoing, but we have done the same experiment with maize plants on the right side of the image. And in this experiment, we observed that in presence of mycorrhizal fungi, the water use efficiency was much higher, and the plants following recovery from a drought stress, so after application of a water stress, when we apply again normal water, well, the plants were able to recover much faster than the non-mycorrhizal plants. This is with maize, and this is now what we want to do with what we are now doing with the wheat. So this is this very short presentation, because now Christophe will show you a film in which the importance of this mycorrhizal fungi is emphasized at the service of, well, I can say, of humanity.
Okay, so let me partager l'écran. So you have to start. Sorry. Okay. Now, can you see it in full screen?
Yes, we can.
Okay.
En 2050, il faudra nourrir plus de 9 milliards d'êtres humains sur Terre. L'agriculture va devoir produire encore plus, mais elle doit aussi s'adapter au changement climatique qui bouleverse ses habitudes. De nouveaux insectes ravageurs ou encore. En 2050, il faudra nourrir plus de 9 milliards d'êtres humains sur Terre. L'agriculture va devoir produire encore plus, mais elle doit aussi s'adapter au changement climatique qui bouleverse ses habitudes. De nouveaux insectes ravageurs ou encore des inondations. Comment relever ces défis qui attendent l'humanité? Je vous emmène à la rencontre de microbiologistes et de bio-ingénieurs qui préparent l'agriculture du futur. Et si tout restait à découvrir?
Avec l'accroissement de la population et le réchauffement climatique, l'agriculture doit faire face à des défis énormes. Il va falloir produire plus et mieux, c'est-à-dire dans des conditions plus durables. Et si je vous disais que les petites billes que je tiens dans ma main font peut-être partie de la solution, qu'elles sont peut-être révolutionnaires pour l'agriculture? Elles ont été mises au point par des chercheurs en microbiologie de l'UC Louvain. Et je peux vous dire qu'ici, l'agriculture durable est prise très au sérieux. Je vous emmène à la rencontre du professeur Stéphane Leclerc qui va nous expliquer un petit peu plus en quoi consistent ces petites billes. Bonjour Stéphane. Alors, est-ce que tu peux nous en dire un peu plus sur ces petites billes?
Alors, ces petites billes, en fait, ce sont des petites structures qu'on a créées au laboratoire et à l'intérieur desquelles on va avoir des micro-organismes. Alors, il faut savoir que ces organismes ont la particularité d'aider le développement des plantes. Donc, lorsqu'on applique ces champignons-là aux plantes, eh bien, elles vont avoir une croissance qui va être améliorée. Elles vont être également plus résistantes à un certain nombre de stress.
Et concrètement, comment on va utiliser ces petites billes sur le terrain?
Alors concrètement, ce qu'on va faire, c'est qu'on va isoler ces micro-organismes, on va les identifier, on va les reproduire en masse et on va les mettre dans ces petites capsules. Et ensuite, ces petites capsules, eh bien, on va les utiliser sur le terrain, par exemple avec des semoirs, sur des tracteurs. Et donc, on a un très grand spécialiste ici de la vie du sol qui s'appelle Hugues Fallis. Et donc, on va avoir le plaisir de pouvoir le rencontrer, qui nous parle un petit peu de l'importance de tous ces micro-organismes, tous ces organismes qui se développent dans le sol.
Alors, spécialiste, je ne sais pas, mais en tout cas, la vie du sol m'intéresse beaucoup dans mon métier d'agronome et d'exploitant agricole. Dans le sol se passe énormément de choses. Il y a une vie qui est extraordinaire. On n'en connaît encore que 5%. Mais en tout cas, cette vie du sol permet de nourrir nos plantes. Ici, on peut voir des légumineuses avec notamment des nodosités dans lesquelles se trouvent des bactéries qui sont capables d'utiliser l'azote de l'air et d'en donner un peu aux plantes. Donc, c'est l'exemple le plus facile à voir, à observer dans nos champs de symbiose en fait entre la vie du sol et nos plantes.
Dans cet échantillon vivent des milliers de micro-organismes différents. Le travail des scientifiques, c'est donc de partir à leur découverte.
Alors Stéphane, nous sommes sortis des champs et nous arrivons maintenant à l'université. Quelles sont les prochaines étapes dans ton laboratoire?
À partir du sol qu'on a prélevé, on va tamiser ce sol, on va isoler les champignons qui se trouvent à l'intérieur du sol, on va les identifier. Et une fois qu'on aura identifié les différents champignons, on va les tester sur les plantes, voir les champignons qui sont les plus efficaces pour ensuite les utiliser et retourner au champ pour améliorer la croissance des plantes.
Qui dit micro-organisme dit taille microscopique. C'est donc un travail minutieux qui attend Monica, rendu possible par des outils de pointe.
Tu peux regarder qu'il y a des spores de différentes couleurs. On a des noirs, des jaunes et un peu transparents. Et du coup, on utilise chacune pour identifier quelle espèce ça correspond.
Bonjour Sylvie. Bonjour. Donc, je reviens d'avoir vu Monica qui m'a demandé de t'apporter ceci. Elle a isolé les spores. Donc maintenant, c'est quoi la prochaine étape?
Eh bien, je vais prendre cette spore et je vais la réassocier à une nouvelle plante afin de permettre au champignon de refaire son cycle de vie et de produire des nouvelles spores. De cette façon, on aura isolé une souche pure ou une espèce pure et on pourra alors l'identifier et réutiliser les nouvelles spores produites pour les expériences au champ ou en laboratoire.
Nous entrons dans la serre collection de la mycothèque de l'Université catholique de Louvain qui abrite des centaines d'échantillons différents provenant des 4 coins du monde.
La plante va rester ici en croissance pendant environ 6 à 8 mois pour que le champignon puisse continuer à établir son cycle de vie, produire de nouvelles spores.
Pour comprendre les interactions entre les champignons et les plantes, les chercheurs font de la culture in vitro. Cela leur permet de visualiser ces échanges et de comprendre comment les deux organismes interagissent. On va aller voir Bérangère qui va nous expliquer tout ça.
Je suis en train d'étudier un peu plus précisément l'interaction plante-champignon. Et donc, comme tu peux voir ici, il y a un transport entre la plante et le champignon. Donc, on a un flux ici du champignon vers la plante. Et dans ce sens-là, on a un flux de la plante vers le champignon. Et donc, on a une interaction et ça permet aux deux de vivre correctement.
Qu'est-ce qui t'a donné envie de venir travailler ici?
J'étais attirée par tout ce qui était nature. Les études, je les ai faites parce que ça permet de toucher un peu à tout, mais aussi d'être sûr d'avoir un boulot varié plus tard et ne pas bloquer dans quelque chose.
Tout d'abord, la biingénierie, c'est vraiment un sujet qui est vraiment très chouette parce que ça nous permet de rencontrer plein d'espèces, plein de mondes dont on n'avait absolument pas connaissance avant, par exemple celui des champignons qui est vraiment fantastique, et celui des plantes également d'ailleurs, et les relations entre elles, parce que la mycorhize, donc l'association entre une plante et un champignon, c'est vraiment quelque chose qui est très récent et dont on ne connaît absolument pas du tout tous les tenants et aboutissants. On ne connaît pas du tout tous les processus biochimiques et moléculaires qui la gouvernent, ni tous les gènes qui sont impliqués, et donc il y a vraiment beaucoup de choses à faire.
Les études de bio-ingénieurs sont effectivement des études qui sont très prenantes parce qu'à la fois elles associent la notion d'ingénieur, donc de découvrir des solutions, de développer des solutions, et la notion de biologie où on s'intéresse au vivant. Donc, il est très important d'avoir des personnes intéressées, d'avoir des outils, d'avoir des financements qui nous permettent d'aller chercher ces micro-organismes-là, qui, pour un grand nombre d'entre eux, peuvent représenter la pharmacopée de demain. Aujourd'hui, on connaît 5% à peu près de la diversité. Ben, imaginez-vous ce que peuvent représenter ces 95% restants pour la santé de l'homme, pour la santé de l'environnement. Et effectivement, on a besoin de vous pour venir nous aider à creuser, à aller plus loin dans la connaissance fondamentale, la connaissance basique de ces organismes au service de l'humanité.
Well, thanks a lot. Again. Can you hear me? Yeah, I guess. So
Yep,
I will now
we
leave the floor to Sarah, and please keep your questions for later on because we have a session of questions and answers. But thanks a lot, Stefan, for your for this talk and this wonderful movie. It gives some ideas for next perspective. Well, I don't know. I'm trying to stop sharing my screen. God.
Yeah, it should just be, I can see if I can ask you to stop sharing.
Because I have so many. Oh. Yeah, okay. I got it done. So, Sarah, are you ready?
Yes, I am on it. So I think you should see now my screen, right?
No.
No?
Yeah.
You do?
I see it, yeah.
Okay, so yeah, hello everyone. I'm Sarah from the Research Institute of Organic Agriculture in Switzerland. And my presentation will give you some insights into the effects of agricultural management practices on soil microbiome, focusing on fungal communities. But first, I will give you a little introduction. And so in recent years, rhizosphere engineering and management became more and more popular as a new research area to enhance agriculture productivity in a sustainable way. In rhizosphere engineering, microbial communities are modified either directly by adding specific fertilizers or nutrients, or by inoculation of beneficial microbes, such as fungi, bacteria, or AMF, just as we have seen before. Or we can manage them indirectly via selected soil management practices, as we have looked into it for the SolACE project for cover crops, crop rotation, or tillage. So one goal of the SolACE project was to improve our understanding of the interaction between crop management practices and crop genotype, especially in the way to improve nutrient use efficiency and drought stress tolerance, but as well as to improve our understanding on below-ground interactions, how microbes are impacted by crop management and genotypes, but also how are they affecting nutrient use efficiency and drought tolerance. So we investigated several field trials, but the results I'm presenting today derived from a long-term field trial in Switzerland, a long-term field tillage trial in Switzerland running since 12 years. And the factors it includes is tillage. We comparing conventional tillage, meaning plowing, and reduced tillage. So just a slow, like a shallow management of the soil. There are three fertilization intensities: non-fertilized, half slurry fertilized, and full slurry fertilized. And we are planting we have planted 4 different winter wheat varieties. So at the time of flowering, we went to the field for sampling, and we sampled whole plants, including parts of the root system and the rhizosphere. We separated the above ground parts to study the biomass and the nitrogen uptake, among others. And the below ground part, we separated the roots from the rhizosphere to study a microbial activity, microbial abundance, and microbial community, microbial diversity and community structure. which we did by DNA sequencing, and what I will focus on the next slides. So to study the diversity of microbes, we can look at alpha diversity. This is the variation of microbes in a single sample. So when we look here at the example, so we have 3 different samples, and the symbols, they representing different species. So it's very in a simple way. And when we look at it, so we would have here, these 2 samples would have a higher alpha diversity than this sample. And then we can look at the beta diversity. The beta diversity describes variations of microbes between samples. So when we have again the same 3 samples, we would have these 2 samples which have the same composition of communities. They are closer together, so they are more similar compared to this sample. So, but now I want to come finally to the results. And here you can see the alpha diversity of fungi in the rhizosphere of the wheat plants, here represented by the Shannon index. This is one index among others to describe alpha diversity. And here you can see the different symbols representing the 4 different wheat varieties under the 3 fertilization intensities, from non-fertilized, half-slurry fertilized, and full slurry fertilized, under the 2 different management practices, so the conventional tillage, the plowed system, and the reduced tillage. And what we can see, or what we observed finally, was that there was a similar diversity on the conventional tillage, whereas on the reduced tillage, the diversity of fungi increased with an increasing intensity of fertilization. And what could be the reason? So, for example, so what we found was that on the reduced tillage, on the full fertilization, we had a higher plant diversity because the plots were heavily infested by weeds. And this could be a potential reason why we have also this higher diversity of fungi in the rhizosphere, because as roots are, the higher the diversity of the plants, the more, which is triggering also the diversity, below ground diversity. And like this, we can have, we can observe a higher diversity of fungi. When we now look at the alpha diversity in the roots, here we observe only a fertilization effect, here with an increasing diversity in the roots on the full fertilization under both tillage practices. And here we can, a potential reason might be that the plants on the full fertilization, they grew bigger, so they have a better nutrition. So they were supposed also maybe to exude more, so to have more root exudate, which is recruiting and feeding a more diverse fungal community below ground. So next I will come to the beta diversity of the fungi in the rhizosphere. So as I explained before, so each dot is representing here one sample, and the closer the 2 dots are, the more similar are the communities. And as you can see here, we have on the left side, we have dots in red and in yellowish. So they are the communities from the conventional, under conventional tillage. And we have here on the right side, the dots in bluish, which are from the reduced tillage plots. And what we can see, we have a very strong separation of these communities here along this x-axis. And in addition, so that means that tillage, that the tillage practice is really shaping the 2 communities, separating them from each other. And what we can see as well is that we have a fertilization effect. We have the full fertilized, the communities under full fertilization are here more on the bottom of the plot, and the non-fertilized here on the top. So also again, as before, the higher plant diversity under reduced tillage might be one reason shaping this community differently, but as well as also that some fungi were already reported to be very sensitive to disturbances, especially like also mycorrhizal fungi. This is a very known factor. And so we might have here specialized fungi which are not present here anymore. And when we now look also at the beta diversity of the fungi in the roots, we can still see a similar picture with the communities under reduced tillage here at the more up the upper part, and under conventional tillage here at the lower part. And also again, like a slight fertilization effect here with the full fertilized more on the left side and the non-fertilized on the right side. But as we see, we have not this sharp separation anymore. The reason might be the same again as we found before, but why we might not have this sharp separation again anymore is that still the wheat might select for similar like fungal families or fungal genera. In addition, what is not very visible on the graph, but what we found statistically, that there is also a slight significant effect of the genotype, which is also makes sense since we know that different genotypes, they have different root exudates, and also as we have heard before, there are differences in the genotypes in recruiting microorganism. Yeah, and this is also is an explanation why we have this effect of genotype in this regard. So what are the next steps? So the next step, this is only as I shown you results from the fungal communities, but the next steps are also to analyze the opuscular mycorrhizal fungal communities and the bacterial communities in the same way we did here for the fungi. And also very importantly, then to link the microbial diversity with the plant performance to see if there are like certain patterns we can see, and to see which might better kind of support the plant in improved nutrient use efficiency, for example. And with this, I want to thank you for your attention. I think I was in time because I was also very trying to stick to it and was afraid to overgo. So
perfect timing, Sarah. Thank you very much.
Thank you.
We'll give the floor to the last speaker of the session, Germain, are you ready?
Yeah, I am. Do you hear me?
Yeah, perfectly.
Okay, perfect. So I share my screen. And I hide you. Okay, so today I'm going to talk to you about a project that was part of SolACE, on which I worked in Montpellier back in 2019. And so as Christophe mentioned at the beginning, this project is not directly connected to plant microbiome interaction, but more focused on plant-plant interaction. Still, hopefully, I think you will find this interesting, and you will see in the end that there is still some follow-up on this project to integrate the plant microbiome interactions. So this experiment was part of the work package 3 of SolACE, which is interested in investigating new agroecosystem strategies and tools to improve crop water use efficiency and nutrient use efficiency. And so one of the strategies investigated in this project is to reintroduce some diversity in our agroecosystem to improve their multifunctionality. As you might know, relying on positive interactions between species and genotype is really at the fundamentals of the agroecological approach. And one a quite straightforward way to do that, to increase diversity, is to mix different varieties of the same species in the same field. And so this is an agronomic practices which is really regaining interest both in the farms and in the labs for the last 10, 20 years. And so the question is this practice of mixing different variety able to improve resource use efficiency in our agroecosystems? So the aim of this experiment was to assess this, to check if varietal mixtures could indeed be a solution to cope with resource-limited conditions, and to eventually identify some traits or trait combination that would help to mix the variety together and to identify some assembly rules. So the experiment was conducted in the platform that Christophe presented you just before, the 4PMI platform. And we used a panel of 36 durum wheat genotypes, which were grown in those transparent rhizotubes that you see on the picture. In each tube, 6 plants were grown together, and the genotypes were either grown as a monoculture with a single genotype in the tubes or as a binary mixture with 2 genotypes in the tubes. And all tubes were replicated in 2 treatments. So one treatment with provided with water and nitrogen, and one treatment with depleted water and nitrogen inputs. And so overall, there were 36 monoculture and 54 binary mixtures, which were replicated in the 2 treatments with 3 replicates per treatment, which was added up to a bit more than 3,000 plants in total. And so here I just put you the timing of the experiment so that you can see that we work here with very young seedlings. The experiment was harvested a bit less than 1 month after sowing, and the different traits that I used in my analysis were measured close to the end of the experiment. So those traits are both above ground and below ground traits. In the above ground compartment, we had access to the number of leaves on the main stem, the number of tillers, and of course the above ground biomass. And below ground, thanks to the image processing capability that is on the 4PMI platform, we had access to different root traits, notably the rooting depth, which is the maximum depth of the root tips on the picture, the root length, the total root length on the picture, and the 2D projection of the root surface. And of course, at harvest, we also had access to the root biomass. I need to mention that for all those root traits, unfortunately, we only had access to the pot level data because it was quite hard to separate the different roots on the pictures. So we have an average value for the whole rhizotube. I jump into the result and show you here the effect of the treatments on the plants. So you have the shoot biomass, root biomass, total biomass, and root to shoot ratio as a function of the treatment. And you can see that the effect of the treatment was quite strong with a reduction in shoot biomass, which was also seen in total biomass, reduction of total biomass, and an increase in root biomass in the water and nitrogen stress treatment, which resulted, of course, in a very strong increase in the root to shoot ratio in the stressed treatment. Accordingly, with what we have seen with the effect on the shoot biomass, you can see that plants in the stress treatment developed less leaves and less tillers. And in the below ground compartments, you here have the 3 different root traits. And you can see that in the water and nitrogen stress treatments, the roots were deeper, longer, and thicker than in the control treatment. So to address the main question of these experiments, I then compared the monocultures and the mixtures. So to do that, I used a very common index, which is a relative yield total. You here have the formula for a binary mixture, such as the one we have in this experiment. So here it's written for a mixture with genotype Y and genotype I and genotype G. And you can see that this index is computed as a ratio between the biomass of the genotype in mixture and the biomass of the genotype in the monoculture. And then you sum up those 2 ratios for the 2 components of the mixture. And based on this computation, the reference value for this index is 1. So if you have an index which is equal to 1, it means that there is no yield difference between the mixtures and the monoculture. If it's higher than 1, you have what we call over-yielding, which means that the mixture produces more biomass than the monoculture. And if the index is lower than 1, it's the opposite. What I show you here is the distribution of this relative yield index for the 3 biomass components over the 54 mixture in the 2 treatments. And what you can see is that on average in this experiment, the mixture were less the mixtures under yield the monocultures by 4% if you look at the total biomass, because the mean value of this index was 0.96. And when we check the effect of the treatment on these indexes to check if the mixture would, for example, yield higher in the stressful condition, we see that there is no effect of the treatment on the relative yield, either on shoot, root, or total biomass. So even if this relative yield index was lower than 1, there was still some variability. If I go back to this slide, you see that there is some variation with some mixtures in which the index is indeed higher than 1 and some mixtures for which it's lower than 1. And so I wanted to try to explain this variability by using the traits of the mixture components as explicative variables. So more specifically for each mixtures, I computed 2 quantities for each trait. The trait mean, which is the average trait between the 2 components, and the trait distance, which is simply the difference between the 2 traits, the absolute difference actually. And so I used the monoculture traits to compute those quantities with the idea to be able to predict mixture performance with the monoculture traits. And then I tried to explain the relative yield on total biomass with all the trait mean and trait distances. So I built a full model with all the traits as explicative variables, and I ran finally a model selection to retain only the most explicative variables. So what you have here is the results for the relative yield total on the control treatment, so without the stress. And here you have all the traits that have been retained by the model selection procedure and their estimated slopes in the multiple linear regression. So what you can see is that in this condition, the traits are really poor predictor of the mixture performance. We have an average R square of 0.07 in the top 10 best models for these conditions. And you can see that those 6 traits, they all have slopes that are close to 0 actually. But if we look at what we have in the stress treatment, we see a quite different picture, because here we have a very strong effect of 1 trait, which correlate pretty strongly with the relative yield total of the mixture. You can see here that the R square is close to 0.5, which is very high. And so this trait is the root surface, which is the projection of the root surface on a two-dimension scale. And so this trait, you can see here that the estimate is negative, is lower than 0, meaning that on average here, the mixtures in which the component of the mixtures had a high root surface were really under yielding their monoculture. So to show you this more explicitly, you have the relationship here between the relative yield of the mixture and the mean root surface of the 2 components in the mixture. So you can see that there is no relationship in the control treatment, but that this relationship is very sharp on the stress treatment, with a negative effect of the mean root surface on the total biomass relative yield of the mixture. So to conclude, we've seen in this experiment that the water and nitrogen stress resulted in an increased allocation of resources to roots in the stress condition, which is something classical in plants that are exposed to soil-related stress. It's been described in multiple species and including at the seedling stage, where it's very important to invest resources in resource harvesting organs when those resources are getting very scarce. We have seen that it seems that there is no benefit of varietal mixtures compared to monocultures if we look at the biomass here, either under optimal or resource limiting conditions. So we have no support for what has been claimed in ecology, the stress gradient hypothesis, which says that positive interactions between different varieties or species get stronger as the abiotic stress increases. We don't see that in this experiment, and it's not the first time these results do not show up in varietal mixture. I put you here 2 examples of studies in which varietal mixtures have been grown in both control condition and stress condition, either for drought or for phosphorus supply. And in those 2 experiments, they had the same results, actually, with no benefits of varietal mixture under the stressful condition. But I need to emphasize that all those experiments are both experiments conducted with plants that were not reaching their maturity. If we look at what happens in the fields, we do have some support for the fact that mixing different varieties has a positive effect on plant performance, especially under low nutrient availability. So this has, for example, been shown in a recent review and meta-analysis by Race and Drinkwater in 2018. And finally, we saw that the traits of the mixture do not predict very well the mixture relative yield in optimal growth condition, with an R-square close to 0.07. So there could be several hypotheses to explain this poor relationship between the traits and the mixture performance. Maybe it was not the most relevant traits to capture and to explain mixture performance. For example, in another study where 20 traits were measured in the field, the R-square was a bit higher. So the traits, since there were more traits, the R-square was higher, but you can see that the more trait is not also the better, because there was like 5, here it's 6 traits or 7 traits, it was 20 traits, but the R-square is not that higher. So it's not all about the number of traits. Maybe another hypothesis is that the variability that is present in this genotype panel is not strong enough for the traits so that we detect any effect of the trait on mixture performance. Or finally, there could be an effect of phenotypic plasticity between monocultures and mixtures, because we do use the traits of the monocultures to try to explain something that happens in the mixtures. And we know that plants can change their phenotype depending on their neighbor. So maybe using monoculture traits, even if it's an interesting approach to be able to predict mixture performance, maybe it's a poor approach if you want to understand the mechanism that explain the interactions between the varieties. And finally, how to explain this negative effect of the high root surface on mixture yields in the stressful condition. So my hypothesis would be that if we have a high root surface in those images, it's mainly related to the root diameter, because we also have the root length as a trait, and the root length did not correlate with this relative yield in the mixtures. So my hypothesis is like those genotypes with thick, I put here thick and long, but I think it's mainly because of the thickness, with thick root systems might exhaust nutrients really fast in the below ground compartments, thus resulting in very strong competitions between the varieties. And so this, in turn, would lead to tragedy of the commons, where the genotypes fight so hard for the resources that eventually they decrease the overall performance of the mixture. So I will end by saying that maybe you will hear again about this experiment and these genotypes pretty soon, because the same experiment was replicated in the field in Montpellier last year as part of the same package. But this time there will be an additional focus on AMF fungi and their diversity. So we'll go from a 2-partner interactions between 2 varieties to a 3-partner interactions with the mycorrhizal fungi in the story, So hopefully this will increase our ability to understand what happens in this mixture. Thank you for your attention.